what I'm going to be doing is I'm going to be talking about some DNA today. So let's do that. Let's talk about some DNA. Opening up the PowerPoint. All right. Let us begin. So, DNA. What is DNA? All right, so DNA is basically our genetic material. And there were numerous people who searched for years before we knew what DNA actually was. So we had Mendel, who discovered modes of hereditary in pea plants in the 1850s. We had Morgan, who found that genes were located on chromosomes in the early 1900s, and then we had Griffith, who did some bacterial work in the 1920s. But this was categorized by transformation, a change in genotype and phenotype due to the assimilation of external substance, DNA, by a cell. So the living S cells are put into a mouse. The mouse dies. The living R cells lag under control, and the mouse is healthy. The heat killed S cells, which are also under control. The mouse is healthy. Mixture of heat killed S cells by living S cells, by the mouse dies. So, living S cells are found in a blood sample. The Avery transformation agent was DNA. Let's talk about the Hershey-Chase experiment. In 1952, it was an experiment with bacteriophages, which are viruses, right here, that infect bacteria. Tested if DNA or protein was the hereditary material in a T2, which is a bacteriophage that infects E. coli, sulfur, S, present a protein, phosphorus, P, is the DNA. The only P was found in the host cell. So it was mixed radioactively with labeled phages with bacteria. The phages infected the bacterial cells right here. 2. Agitated in a blender to separate phages outside the bacteria from the bacterial cells. So the empty protein shell, the phages were another DNA. 3. Centrifuge the mixture so that bacteria formed a pellet at the bottom of the test tube. 4. Measured the radioactivity in the pellet and the liquid radioactivity. The phage protein right here was in the liquid. Now batch 2. Phages were grown with radioactive phosphorus, which was incorporated into phage. DNA, blue. DNA structure. All right. We had Chargraph, who discovered the ratio of the nucleotide bases, A to T, and C to G. The structure of DNA was researched by Pauling and Wilkins and Franklin. Watson and Crick in 1953 proposed the structure of DNA after viewing an X-ray diffraction photo by Rosalind Franklin. The double helix. Nucleotides are nitrogenous bases, thymine, adenine, cytosine, and guanine, sugar, deoxyribose in the phosphate group. Basic pairing rules. All right, so purines are adenine, then guanine. Pyrimidines, C and T, are cytosine, and thymine. Adenene's H plus bonds 2 times with bonds twice with T, and with thymine and cytosine's H plus bonds 3 times with a guanine. Van der Waals attraction between the stacked pairs. DNA replication. Watson and Crick's strands are complementary. The nucleotides line up on template according to base pair rules. The parent molecule has 2 complementary strands of DNA. Each base is paired by hydrogen bonding with its specific partner, adenine to thymine and guanine to cytosine. The first step in replication is separation of the 2 DNA strands. Each parental strand now serves as a template that determines the order of nucleotides along the new complementary strand. The nucleotides are connected to form the sugar phosphate backbones of the new strands. Each daughter DNA molecule consists of 1 parental strand and 1 new strand. 3 replication models. All right. And the first one, which is the conservative model. The 2 parental strands reassociate after acting as templates for new strands, thus restoring the parental double helix. Semi-conservative model. The 2 strands of the parental molecule separate and each functions as a template for synthesis of a new complementary strand. Dispersive model. Each strand of both daughter molecules contain a mixture of old and newly synthesized DNA. Melton and Stahl concluded that DNA follows the semi-conservative model, which is very, very true. It does. DNA replication in action. All right. Origin of replication, which is the bubbles equal beginning of replication. Replication fork, Y-shaped region where new strands of DNA are elongating. The helicase catalyzes the untwisting of the DNA at the replication fork. DNA polymerized catalyzes the elongation of new DNA. So here's the bubble, here's the replication fork where the parental strand and the daughter strand meet, and 2 daughter DNA molecules. All right, anti-parallel nature. The sugar phosphate backbone runs in opposite directions thanks to Crick. 1 strand runs 5' to 3', while the other runs 3' to 5'. DNA polymerase only adds nucleotides at the 3' end, forming new DNA strands in the 5' to 3' direction only. So this is very important to understand. DNA polymerase only adds nucleotides at the 3' end, forming new DNA strands in the 5' to 3' direction only. So this way, not this way, only the 5' to 3'. Leading and lagging. The leading strand synthesizes towards the replication fork only in a 5' to 3' direction from the 3' to 5' master strand. So it goes this way. See, 5' to 3' and it creates new at the 3' end, only at the 3' end. The lagging strand synthesizes away from the replication fork in small pieces, Okazaki fragments, joined by a DNA ligase. The lagging strand. Primase enzyme attaches a primer, a short sequence of RNA. Sometimes it can be DNA. Small segments are again they are only replicated from 5' to 3'. After the strand replicates, the primer falls off. Helpful proteins. There are numerous helpful proteins. Helicase is a protein. Unwinds parental double helix at replication forks. Single-strand binding protein. Binds to and stabilizes single-stranded DNA until it can be used as a template. Topoisomerase. Correct. Overwinding ahead of replication forks by breaking, swiveling, and rejoining DNA strands. Function for the leading strand with primase synthesizes a single RNA primer at the 5' end of the leading strand. The function for the lagging strand synthesizes an RNA primer at the 5' end of each Okazaki fragment. DNA polymerase III continuously synthesizes the leading strand, adding to the primer. Elongates each Okazaki fragment, adding onto its primer. DNA polymerase I removes a primer from the 5' end of a leading strand and replaces it with DNA, adding it onto the adjacent 3' end. Again, DNA is only added onto the 3' end. Removes a primer from the 5' end of each fragment and replaces it with DNA, adding onto the 3' end of the adjacent fragment. Again, DNA is only added onto at the 3' end. DNA ligase joins the 3' end of the DNA that replicates the primer to the rest of the leading strand. Joins the Okazaki fragments. Proteins in action. Alright, this is a really good diagram. So, in stage 1, the helicase unwinds the parental double helix right here. Number 2, molecules of single-strand binding protein stabilize the unwound template strand right here. 3, the leading strand is synthesized continuously in the 5' to 3' direction. 5' to 3' direction by DNA polymerase III. 4, primase begins synthesis of the RNA primer for the 5th Okazaki fragment. DNA polymerase III is completing the synthesis of the 4th fragment. When it reaches the RNA primer end on the 3rd fragment, it will disassociate and move to the replication fork and add DNA nucleotides to the 3' end of the 5th fragment. Primer again, only added on the 3' end. DNA polymerase I removes the primer from the 5' end of the 2nd fragment, replacing it with DNA nucleotides that adds one by one again to the 3' end. Only added on again to the 3' end of the 3rd fragment. The replacement of the last RNA nucleotide with DNA leaves a sugar phosphate backbone with a free 3' end. DNA ligase bond to the 3' end of the 2nd fragment, to the 5' end of the 1st fragment. Proofreading. Let's go back. I'm sorry about that. Mismatch repair DNA polymerase. Excision repair nuclease. A thymine dimer distorts the DNA molecule. A nuclease enzyme cuts the damaged DNA strand at 2 points, and the damaged section is removed. To repair synthesis by a DNA polymerase fills in the missing nucleotides. 4. DNA ligase seals the free end of the new DNA to the old DNA, making the strand complete. Shortening ends. Because lagging strands need 3' end, replication cannot extend to the very end of a DNA strand. Chromosomes contain telomeres, which are repeating sequences of DNA. However, they do not repeat genes. They do not contain genes. Telomerase lengthens the telomeres so that they can't continually get shorter. Chromosomes can divide without losing genes. And that was it. Thank you for your time, and have a great day.